All right. Well, welcome everybody to Plato Frontiers in Life Sciences. Great to see everybody. Thanks for coming. Our speaker today is Aaron Simmons. I'm going to ask Aaron the 5 questions that I ask everybody. Aaron, where were you born?
Born in Kansas City, Kansas.
Hey, and where'd you go to high school?
Went to Boswell High School in Fort Worth, Texas.
Wow, you moved.
Yeah.
Where'd you go for your undergrad and what did you study?
I went to the University of Oklahoma, and I studied chemical engineering.
And then where'd you go for your advanced degrees and what did you study?
So I actually did an accelerated master's at the University of Oklahoma, and then I'm currently doing my PhD here at the University of Wisconsin, all in chemical engineering. This department is chemical and biological engineering.
Very good. So today you're going to talk with us about a multi-omic approach to improving the production of human pluripotent stem cell derived cardiomyocytes. Let's hear it for heart cells. All right, thank you, Aaron. And go ahead, I'll be in the back from time to time. But please
Okay,
start.
great. Thanks a lot. Thanks for the introduction, and thanks for inviting me to come speak to you all today. I realize this is a fairly involved title, and hopefully for the first few slides that I'll be going through, I'll kind of break this apart and explain each of these parts of the title here. As I mentioned, I'm from the Department of Chemical and Biological Engineering here at the University of Wisconsin. A lot of the work that are in the Palachuk lab, a lot of the work that we do is also in conjunction with a engineering research center funded through NSF, which the logo's over here, is particularly for cell manufacturing technologies. And the whole goal of this center is to try to improve the production of different stem cell or cell-based therapies for kind of novel regenerative medicine applications.
And Aaron, the little slide is showing up on the YouTube and on the Zoom. Is there a way to adjust that, or is that how
The little slide is showing up?
Yeah, your next slide is showing up.
Oh, the slide presenter show?
Yeah.
I can just, instead of duplicating, let's see. Oh, that's not what we want. There we go. Does that work better?
It hasn't changed yet. I'm going to zoom in. Now it is.
Okay.
Very
Let's
good.
try and
All
reconnect
right,
with
thank
this.
you. Apologize for the.
No worries. Out of the way. Okay. Good to continue.
Yes, please.
Okay. So briefly, the talk that I'll present to you guys today is we'll kind of motivate why we're interested in this particular cell type. I'll introduce what the cell type is and how we produce them. Kind of an initial motivating experiment that I performed before we initiated this large project that I'll be talking about today. Go through a few different aspects of this particular project, and then kind of close with ongoing and future work. I will mention this is still an ongoing research effort. Yes.
You have to start screen sharing.
Is it not screen sharing again? Is that working now?
Now we're looking a little bit.
Okay.
I apologize.
No worries.
Sorry for the folks at home watching.
Okay. As I mentioned, this is an ongoing research effort, so I'll be showing some of the initial findings that we're starting to glean from our experiments and how we're approaching the future analyses. On the right here, we see cardiomyocytes. Cardio meaning heart, myo meaning muscle, and cyte meaning cell, are basically the functional cells of the heart that produce the beating contractions that allow you to pump blood throughout your body. These are a highly energetic cell type. So you can see here, this is with a calcium indicator dye, where you can see the cells actually beating and as they rapidly propagate calcium through them to actually affect the function of the cells. So I mentioned I'm going to start with the motivation. So the biggest motivation here is heart disease. This is the biggest killer in the US and worldwide, with approximately 1 in every 3 deaths attributed to heart disease. Annually in the US alone, there are approximately 720,000 people that suffer myocardial infarctions or heart attacks. And there's a heart disease associated death approximately every 60 seconds in the US alone. This leads to significant economic burden and numbers that are predicted to rise quite substantially over the next decade or so. And so one of the big things that you get often with heart disease is you'll get a myocardial infarction or a heart attack. So on the top here, we have a normal heart where you can see, good muscle tissue morphology. After you have a heart attack, which is essentially loss of oxygen to the heart, you have a significant area of cell death because there's no oxygen able to flow to that region of the heart. This eventually leads to pathologic remodeling. So you have the ischemia, which is the lack of oxygen, which leads to left ventricular specifically. So this is the left ventricle of the heart. Cardiomyocyte death can lead to ventricular wall thinning and remodeling, a lot of scar tissue formation in this region, and overall decreased cardiac output. So your heart isn't able to pump as effectively as it was before. This can eventually lead to heart failure and death as well. And overall, this decreases the health span, so the number of healthy years that a person has after having the heart attack, and eventually decrease lifespan as well. So there are many different current treatments out there on the market today. So there's a lot of pharmacological interventions I'm sure many of you have heard of, so beta blockers, ACE inhibitors, and diuretics to try to help pump blood better. Surgical interventions such as cardiac patching, some meshes, trying to actually go in and sculpt and reshape that heart where you get thick scar tissue formation, or even just a whole heart transplant. There's also device-based interventions, so such as pacemakers to try to help provide additional beating excitation to the heart, defibrillators, and then left ventricular assist devices shown here that really just tries to provide additional pumping to that region of the heart that is no longer pumping efficiently. But really one thing all of these have in common is they're really just treating the symptoms and not the underlying disease or the underlying lack of cardiac output. So they're trying to mitigate the problem of decreased cardiac output without actually trying to restore native heart pumping efficacy. So what we seek in our lab and in the field is kind of a regenerative cell-based intervention. We're particularly focused right now on stem cell-based interventions, where we can take stem cells and make these cardiac cells to then implant into the heart. So this has been shown before. Didn't realize this is cutting off part of the screen. So this has been shown before in animal models all the way up through non-human primates, where in here, these researchers took human stem cells, human pluripotent stem cells, which I'll describe in a moment. They differentiated them into these cardiomyocytes, and they induced a myocardial infarction or a heart attack model. And in this particular study in cynomolgus monkeys, these cells were GFP positive, so they have green fluorescent proteins. So they're showing up in green on the figure here. I apologize that it's red and green if there's anyone red-green colorblind. I did not make this figure. But we do see after 2 weeks after the myocardial infarction, they implanted these stem cells, and then they assessed them later on. They showed that they did engraft, as here on the left with the green regions. Go to the next slide. To the next. No. Yes.
Did those researchers also look at whether the cardiac output improved?
Yes, so yeah, that's actually the next slide I'm looking at right here. So two of the predominant things that we look at in the field for looking at heart efficacy or the ability of the heart to function are called the ejection fraction. So the amount of the volume that the heart is actually able to output. So in every pump, how much of the total volume of that chamber is able to be expelled into the venous system. And so we see an improvement in ejection fraction. So we have pre-treatment, so after the myocardial infarction, but before they administered either their stem-cell-derived cardiomyocyte treatment or just a control model that they did not add the pluripotent stem-cell-derived cardiomyocytes. And then in green here, I've added what a healthy control is expected to be for these different metrics. So here we saw these researchers saw improved ejection fraction, nearing what we expected for a healthy control, and improved fractional shortening, which is essentially how much the individual cells are able to contract. So they like to be able to contract more to be able to produce more pumping efficacy. So now I'll briefly talk about, we've kind of shown the motivation here, and I'll kind of talk about some of the different words in the title here. So the first, I'll talk about human pluripotent stem cells. So human pluripotent stem cells, or HPSCs, are defined, first I'll define this word pluripotency, and all that means is that it's able to become any cell type in the body. So here, if you remember all the way back to kind of some developmental biology classes or some intro biology classes, you can break up your cells into essentially 3 different germ layers that form initially. You've got the ectoderm, the mesoderm, and the endoderm, and these give rise to essentially all cell types within the body. So these pluripotent stem cells are able to become anything from neurons to blood cells and everything in between. Of particular interest for us is the cardiomyocyte. And they come in essentially two different forms. We've got either ESCs, which are embryonic stem cells, which are actually derived from a developing blastocyst. These were originally discovered, how to culture these by Jamie Thompson's lab here at the University of Wisconsin, typically from discarded in vitro fertilization embryos. But these obviously have a lot of ethical concerns and other problems with procurement and things like that. So since then, the field has developed what are called induced pluripotent stem cells, where you can take a terminally differentiated cell type. So for example, a blood draw, or you can take a couple of cells from your skin, and you can essentially reprogram these back to an embryonic-like state. So these cells that were once skin cells can then become essentially an embryonic-like cell, and then now they can derive into any of the other cell types in the body again. So it's a very powerful technique. This was also pioneered by Jamie Thompson here at the University of Wisconsin, in parallel to another researcher, Dr. Yamanaka in Japan.
What made you think that we could do that?
Like me, this was before I was ever in the field. But essentially what they did is they went through and they took, they did a lot of these next-gen sequencing, things like, so they looked at a lot of proteins and RNAs that are upregulated or that are overexpressed in an embryo cell or in these embryonic stem cells versus other differentiated cell types. And they found a core set of proteins that look like they have high upregulation to be able to maintain this state. And they essentially just take those. So once they found that, then they did a lot of experiments to add these different proteins to the cells exogenously, so they can add them in, or they can try to use some newer technologies to get these to be expressed within a terminally differentiated cell. And they found that they were actually able to reprogram back to this embryonic-like state. So it was a lot of discovery-based work on just looking at the differences between an embryonic stem cell and any other terminally differentiated cell. And I think it took, millions and millions of dollars and many, many years to actually be able to efficiently generate these. I will say that they were generated a little bit earlier in other animal systems, such as mice and things like that. Although, interestingly enough, the biology didn't port over exactly. So the factors that are often used in mouse embryonic stem cells don't all work as well to produce human pluripotent stem cells, for example, just some differences in the underlying biology though. Does that answer your question?
Kind of. It just seems to me like an amazing feature that how would you suspect that?
Yeah. Yeah, it's a very interesting finding. It actually goes back to some earlier hypotheses by earlier researchers. So there's something called the Waddington landscape by Dr. Waddington. This was in the mid 20th century, I think, where he hypothesized that there's essentially this underlying landscape to all the cells and how they become different cell types in the body. And essentially, what they were able to, or what he hypothesized was that there theoretically could be a way to push cells back up this into a less, or there's essentially like a hills and valleys kind of model where your cell can go from the top, which is your pluripotent cell, and it can drive into different cell types depending on different signals it receives to get down. And this is the underlying landscape. So then researchers thought, well, maybe we could try to push something back up that. And that's essentially how they ended up finding, they just looked at the profiling differences between these two cell types, or between these and all of these. It was very interesting for the field, and it's definitely exploded a lot of the research in pluripotent stem cells. So the main two takeaways here for pluripotent stem cells, they have essentially an indefinite self-renewal potential, so they can continually produce more and more of themselves, which is really good when we're looking at trying to make large batches of these cells. So you can take essentially one pluripotent stem cell, and you can make billions and trillions of cells from that. And then each of these can then become different cell types as well. So then they also have the ability to differentiate into any cell type in the body, thus this pluripotency term. The only limitation is they're not able to become extra embryonic tissue, such as placenta and things like that. That's one level higher in potency, called a totipotent cell, which you can actually derive from an earlier stage in the embryo, kind of the 2 or 4 or the 4 or 8 cell mass stage. And now, so I've mentioned a little bit how we can make these pluripotent stem cells, and then how do we actually take these all the way down specifically to the cardiomyocytes, or our cell type of interest. So a protocol developed in our lab almost a decade ago now. Basically, we try to follow the standard developmental trajectory. So exactly how the heart would develop in the human body, we try to mimic that. And so we start with our induced pluripotent stem cell or embryonic stem cell, you could. And just through modulating this one particular pathway called Wnt signaling, the details of which are important here, we can activate Wnt signaling to drive this naive stem cell into a mesoderm or an endoderm, kind of an intermediate between mesoderm and endoderm, one of those three germ layers. And then we can inhibit Wnt signaling to try to specify this to a specific subset of mesodermal tissues called cardiac mesoderm. We can then further, by adding insulin, try to drive this to a cardiac progenitor and eventually down to a cardiomyocyte. There are a few markers that are known from developmental biology that are reminiscent of each of these different cell types or that define each of these cell types. But there's really not much known about a lot of these intermediate states, and particularly if there's anything that can distinguish between a mesoendodermal cell that will become a cardiomyocyte versus a mesoendodermal cell that will become a bone cell, for example. So the overall kind of applications and limitations of this technology so far, I've already mentioned kind of the cell-based therapy approach, which can actually be bifurcated into either autologous cell-based therapy, wherein the donor, so the one from which you originally derived the cells, so you have a donor over here, you can take your cells, either a fibroblast, which is a skin cell, or a blood draw. You can then again reprogram this into this pluripotent cell type state and then differentiate it into a cardiomyocyte. This cardiomyocyte can then be used, as I mentioned, in a cell therapy application, either autologous, where it goes into the same person from which you derived that initial donor tissue, or into an allogeneic transplant, so it can be implanted into additional patients. Particularly for our field and for cardiomyocytes, a lot of the work is trying to develop allogeneic therapies, wherein you could derive cells from one patient and potentially treat hundreds or thousands of patients from that one patient, again, due to the vast expansion potential of these pluripotent stem cells. There are other different applications as well, such as drug screening. One of the biggest causes of drugs failing clinical trials is off-target organ toxicity, and predominantly in the heart or the liver. So being able to have a platform to in vitro or in the dish screen drugs for potential problems in the heart before you ever get to clinical trials can help save a lot of time and a lot of money and a lot of problems for those clinical trial patients. Also disease modeling. So there's a lot of diseases, particularly congenital diseases, that are known to affect the heart early in development. And so you can actually take stem cell lines derived from people with different diseases, and you can derive these into cardiomyocytes and then do some different assays directly on those instead of having to get patient tissue. And finally, for kind of standard developmental biology applications to better understand the underlying biology of the system. Most of the work, all the work that I'll talk about today, and most of the work of the emphasis in our lab is really agnostic of the upstream derivation of the pluripotent stem cell, either embryonic or induced pluripotent, and agnostic of the downstream application. We really just focus on trying to improve this process and be able to scale this process up to large production scale, and particularly with a focus on quality assurance and quality control metrics. So being able to make sure that we're differentiating well along this lineage and improve the overall process.
When you're implanting in other patients, is there any problem with rejection?
So there's a few different ways the field is trying to approach that right now. One are through just kind of standard donor matching, so similar to like a blood HLA matching. So you make sure that someone has the same blood type. So you would derive cells from people with different HLA profiles or human leukocyte antigen blood type profiles, and then make sure you'd have basically several batches for each of those. So when a patient came to you, you understand their profile, and then you'd use only cells that were derived with that particular profile. The other way that it's being utilized and being studied quite a bit right now are through using what are called kind of universal cell technologies. So they try to knock out specific HLA phenotypes and HLA antigens and try to upregulate other immunosuppressive molecules on these cell types to be able to evade that mismatching problem. So that you could theoretically use one batch of a cell that had been genetically modified in any patient. It was a very, very good question, and very interesting work going on right now. Most of the work right now, and I guess I will mention there are also clinical trials coming on board right now in Japan and soon to be, I think, in Europe as well, for stem cell-derived cardiomyocytes in human patients. I mean, they're all using HLA matching, though, I believe, right now. So overall, in this,
Why are they ahead of us?
yeah, that's a good question. I don't really know the answer to that. I think some of the regulatory mechanisms they have are a little bit different than ours. And also a lot of the seminal work has been done in Japan as well. So I think they kind of hit us a little bit ahead of the curve on some of that. Also, some of the IP considerations. So the original IP for making these pluripotent stem cells is actually developed in Japan. So I think they might get a little bit ahead of us on that as well. So overall, in this green square where we find ourselves on this differentiating from stem cells to cardiomyocytes, there are several current limitations in the process. So there's batch-to-batch variability. So if I take a batch of stem cells and I differentiate them today, and I use the exact same protocol, and next week I use the exact same cell source, the exact same protocol, I can get drastically different results. So today I might get a really high purity of cardiomyocytes. So maybe 95% of my cells become my cardiomyocytes. And then a week from now, I do the exact same process, and I think I'm doing everything exactly the same. And maybe I only get 10% of my cells are cardiomyocytes, and 90% are off-target cell types. So that's a very big problem in the field. Cell line to cell line variability. So I mentioned there's different types of, I don't know why that's popping up. There's different types of, there's different cell lines. So you can derive these induced pluripotent stem cells from many different patients, and those have significant variability as well. So if I take an iPS cell derived from one patient, and I take an iPS cell derived from another patient, those can actually differentiate very differently into the cardiomyocyte. So sometimes you even have to change your protocol. So instead of being able to use the exact same protocol every time, did it stop sharing?
Yeah, can you restart screen sharing? For whatever reason. It bumped
So
up.
many problems.
Yeah, I've never had a problem with Zoom before.
Okay, are we back up?
Yes.
Okay, so cell line to cell line variability. Again, one patient's cells and another patient's cells can differentiate very differently. And finally, and then also lack of methods to monitor the progression. I showed before there are a few markers that we can look at kind of macro scale lineages. So we can look at a mesodermal cell type or different intermediates, and we can kind of get an idea that it's at least become the right intermediate, but it doesn't really predict whether or not we're on the correct trajectory all the way to a cardiomyocyte.
Might it make a difference if you took it from the skin or from the blood?
So there actually are some very interesting studies that look at the initial tissue source that you derive the cell from. And they do show some bias in the different lineages that you can become. So some researchers have even gone so far as to take skin from a cardiac biopsy. So after you take a small piece of heart, reprogram that back to an induced pluripotent stem cell, and they find that those can actually more preferentially become cardiomyocytes. There's some other different, there's underlying what's called epigenetic modifiers that they think are kind of a memory that's retained within the cell, basically modifying how the genome is oriented, how it's packed into the cell, into the nucleus, and that can actually affect how well that cell might become a different cell type. So there's very interesting work on that. And that can lead into some of the cell line to cell line variability potentially.
Sorry, I have a quick question. Could you speak on the batch to batch variability? Do we know why that could be the case?
We don't know why that's the case really right now. And that actually motivates the next click on my slide here.
Amazing. Thank you.
Sorry. The question was, can you speak to the batch to batch variability? Do we know why that is? And the last slide, the last thing here is immature cardiomyocyte phenotypes. So we actually derive these cells, and they more resemble a fetal-like cardiomyocyte than a fully adult mature phenotype. So they look very different. So they're much, they can't quite output as much force, and they're not as large and metabolically active. Is this closing out again? I'm gonna have to relaunch again.
Yes.
Okay. Sorry, everyone. I've never.
Well, it might be the Wi-Fi here. That's why.
Oh yeah. Once it gives me the option to reconnect. Because it's not showing up right now.
Not yet.
Okay. Any other questions while we wait for this to?
Why
Relaunch.
not simply take the person's own cell and go through the whole process of deriving?
So on the autologous approach?
Yeah.
Yeah, so there are one of the big benefits to doing an allogeneic approach or using one donor to many different patients, is you get economies of scale. So it's a lot, by doing large batches, you can actually get your price per dose down much, much lower than having to do individual runs for everything. So if you're familiar at all with some of the recent work in CAR T therapies, I'm not sure if people are familiar with that. Those are all currently done in allogeneic fashion. So they take one person's cells, they genetically modify them and implant them back into that same patient. Those therapies cost on the order of around $500,000 for one therapy. And that's because every single patient has their own specific batch. And so you get a lot of problems with, it takes a lot more handling of that tissue, of that particular specimen. And basically you need isolated chambers for everything to be done. Whereas if you can do a large scale production, you get, as I mentioned, these economies of scale, and your price per dose comes down quite significantly. So it's really kind of more towards trying to make these more cost effective so that, the general population will be able to achieve it instead of, billionaires that can afford dropping $500,000 on one therapy. Okay, I think we're back up. So these top three, the variability, cell line to cell line variability, and lack of methods to monitor progression really come down to an incomplete understanding of the overall developmental biology and the overall process of deriving these stem cell-derived cardiomyocytes. And that in turn kind of closes the loop and leads to poor process robustness. So because we don't understand the process completely, we don't really know what might be going wrong in the process. And that's really what we're trying to intervene. As I mentioned, I can do the exact same thing today and tomorrow, but there could be minor differences that I'm not aware of. Maybe the humidity is slightly different and that somehow has an effect. Or maybe my time I'm off by, a few minutes or something in a particular procedure. And there could be some underlying variables that we're not capturing or we're not aware of that could be drastically altering this process. So overall,
probably the moon phases.
it could, yeah, could be. So overall, we're looking to try to optimize this stem cell-derived cardiomyocyte production through development of scalable methods. As I mentioned, for these allogeneic therapies, we'd like to be able to do this at scale so we can get those economies of scale and get that pricing down in these large batches. But scalable methods to be able to monitor the development of these cells, so as they differentiate through this process, and then improve the overall process robustness. So kind of the overall project vision here, again, we have the differentiation from our stem cell to our cardiomyocyte. And current process knowledge leads to little ability to be able to predict a priori if you're going to become a cardiomyocyte. In that you don't know until you have cardiomyocytes if you're going to get cardiomyocytes. So what we'd like to be able to do is be able to basically push that much earlier in the process and be able to continue earlier and continually predict the final cardiomyocyte purity. So be able to see, as early in the process as possible. Are there specific markers that we can be looking for to make sure that we're becoming a high purity differentiation versus a low purity differentiation? Such that if you know you're going off spec and you're becoming a low purity differentiation and you're not going to achieve a high amount of cardiomyocytes, you can either fail the batch and say, I'm not going to waste that extra 2 weeks to make this batch that I know is not going to work. Or you could design methods to intervene and say, okay, well, I know it's going off in this particular manner. I'm going to intervene in this particular, by perturbing the system in this way to try to bring it back onto a high specification process. Does that make sense? Mm-hmm. And then finally, I'll talk about multi-omega. What do we mean by this? So I'll bring you back to your early days in school with the central dogma of molecular biology. So if you remember way back when, DNA gets transcribed into mRNA, which then gets translated into proteins. And this is called the central dogma of molecular biology. And these proteins, through enzymatic activity, can essentially interact with small molecules or metabolites within the cells to affect the overall change in that as well. But this is kind of the central dogma, but there's a lot more complicated things going on here. So proteins can actually directly interact with not just metabolites, but mRNA. They can affect their own translation. They can affect transcription, and they can actually modify DNA itself. Metabolites can do very similar things. So they can modify proteins, they can modify mRNA, and they can modify DNA, all kind of regulating each step of this process. And it's these together, this complex interactive environment that altogether leads to the final cell structure and function. So with multiomics, we're trying to understand all of these different individual techniques or these individual levels of the regulatory system. With conventional molecular biology, you might study one gene mutation. So you could say this particular patient has a particular gene mutation that leads to some disease. And you can say, okay, how does that affect the mRNA expression of different particular transcripts in the cell? How does that affect protein regulation, and how does that potentially affect, maybe one or two or a few metabolites? Omics, what that does is essentially trying to look at everything at any of those individual levels. So there's something called genomics, where you look at like the entire genome to see what's different over time, or what's different within the genome between patients. Transcriptomics is looking at all the transcripts or all the mRNAs within the cell to try to see what's going on across the board, kind of a discovery-based approach. You don't know beforehand what you're looking for, you're just trying to see what you see. Similar for proteomics and protein levels, and metabolites, you have metabolomics. And really this multiomics integration is we're looking at transomics. So we're trying to understand better that complex interaction between these different levels of regulation. So I showed on that previous slide where you can have metabolites that can affect proteins and all the way up. And basically all of these are interconnected with each other. And we're trying to better understand this entire process to provide better understanding and better overall meaning to what's going on here, so that we can try to have a more complex understanding and be able to actually intervene in meaningful ways. Does that make sense to everyone? So I mentioned I'll go to a proof of concept experiment that I initially performed probably over 2 years ago now. So we first asked the question, how or do key metabolic pathways change during the differentiation of stem cells to cardiomyocytes? So obviously there are certain pathways, we know a lot about stem cells, we know a lot about cardiomyocytes, but we don't really know, or wasn't really known as well before what's going on in the changes here, particularly in metabolic pathways. So we wanted to see, are these actually changing? We know the starting point and the ending point are different. We don't necessarily know how they change. Is it just a linear change over time? If you have something highly expressed in cardiomyocytes, lowly expressed in stem cells, is it just linearly changing over time, or are there more complicated dynamic processes going on here? So we did here, and I'll walk you through this a little bit more. I went through and designed specific assays to look at particular mRNAs or particular transcripts within cells that we collected at different times during this differentiation process. So we developed a few different assays to look at particular proteins involved in kind of glucose signaling or glucose metabolism. So looking at glucose uptake into the cells, storage into different forms within the cell, and then down through glycolysis. We also looked at fatty acid metabolism. So fatty acids are heavily utilized by mature cardiomyocytes, and we expect cardiomyocytes to be more reliant on this than stem cells would be. So we looked at a few different proteins involved in this process. Looked at a few different proteins in the citric acid cycle or the Krebs cycle as well. We expect this to be upregulated in cardiomyocytes versus stem cells, since they are a more energy demanding cell type. Similarly, looked at a couple of different proteins in the electron transport chain. Again, these are highly energy demanding cells. We'd like to be able to look at and see if they are upregulating pathways that we would expect to be upregulated in the cardiomyocytes. Another key thing that we expect in the cardiomyocytes is there's this creatine shuttling pathway that is, so your cells like to contract a lot, and it requires a lot of energy to contract, and that energy comes in the form of phosphates. And so the way this is actually achieved within cardiomyocytes is through these two proteins here, which are called creatine kinases. And they essentially take the high energy phosphate from an ATP, and they transfer it out of the mitochondria, and then they all the way down to the sarcomere, which is the functional unit of the cardiomyocyte, to allow it to contract. So we expect these to be upregulated in the cardiomyocytes as well. But again, we wanted to see how that happens in the process. Is it early in the process? Is it late? Or is it how are the dynamics involved there? And finally, looking at a few different master regulators of metabolism. So these are a couple of different proteins that have basically opposing effects on metabolic activity within the cells. And then a few different proteins involved in mitochondrial dynamics and overall mitochondrial content within the cells. Again, because cardiomyocytes are very heavily metabolically active, they produce a lot more mitochondria. So we expect that to be upregulated at the end time point, but we want to see how that changed over time. So now we've kind of looked at a few, all in green here, I've got essentially different transcripts that we were looking at over time in this process. So I'm not going to go through this in huge detail, but just kind of the long story short here is we didn't just see kind of a linear increase or linear decrease in these over time. We saw quite dynamic trends throughout the differentiation. So what we're going to do here on the x-axis is the day of the differentiation. So day 0 is we're a stem cell, and by the end we're a cardiomyocyte. And these are going through the kind of those intermediate intermediate stages through, mesoderm, cardiac mesoderm, so on and so forth. And on the y-axis here, this is a typical scaled expression level for these different transcripts. So we see there's a number of these that kind of abruptly turn on at this early phase in the differentiation, or kind of midway through the differentiation. We see some other ones that show kind of this other interesting dynamic trends where they tend to turn on at this same stage in the differentiation, but then start to trail off as they finish their differentiation through the cardiomyocytes. And then other ones that express much more dynamic trends. So, for example, this one here and SLC2A1, which includes one of the glucose transporters. We see this very interesting scheme over time where we have a drastic decrease in it and an increase and then a decrease again. I'm not going to explain this too much more, but just the initial experiment here was to show, are there differences over time and how are they changing over time, to kind of show that we might have success in looking at this in more detail. Obviously, if we don't see any changes over time at that level, then we can't expect that we're going to see any changes at other levels. So that's all looking at temporal phenotyping, or looking at the differences over time. So again, that was just from the stem cell to the cardiomyocyte. We'd also like to bring in discriminatory phenotyping. So one of my initial slides, I was saying, we want to be able to tell if this is going to become a good differentiation or a bad differentiation. So again, a low purity of cardiomyocytes or a high purity of cardiomyocytes, as determined here by flow cytometry, looking at a specific cardiac marker that we use quite often. And then we'd like to do that, as I mentioned, in the multiomic side of things. So looking all the way at the RNA or the transcript level, the protein level, and metabolite levels. So in this process here, essentially at intermittent time points throughout the differentiation, which is actually 8 time points, essentially every other day in the differentiation, we collected parallel wells for trisol extraction, or for extraction to be able to do RNA-seq for looking at all the transcripts, transcriptomics. We're actually still waiting on this data to come back. So I don't have anything to speak to that. Hopefully we should have the data back in the next couple of weeks and have some interesting results from that. The other data that I will be talking about today is we took a parallel well and we took the media from that to do what's called metabolic footprinting, which I'll explain in a few slides, which you can use a technique called nuclear magnetic resonance, or NMR, to be able to look at basically the metabolites that are present within the cell culture media. We can also take the remaining cells. So this is just taking basically all the food that's on top of the cells. And we can take the cells themselves and we can extract all the metabolites that are within the cells. So here we're getting the metabolites that are outside of the cells. And here we're taking the metabolites that are within the cells. And we can run that on a specialized mass spectrometer, which I'll explain here in a little bit as well. And then we can take the protein pellet itself. So this is looking at the metabolites, and then we can take the proteins from this exact same set of cells, and then we can perform a proteomics assessment on these. And I'll explain each of these techniques as we get to them. And then for this particular data set that I'll be sharing with you today, we did this for batches of differentiations that resulted in a high purity of differentiation, here defined by 75+% of cardiomyocytes, and a differentiation that resulted in bad or in poor quality or poor purity. Here we have a batch that was 35 ± 3% cardiomyocyte purity. If you'd like to learn more about this method, this multiomics method, we actually have a paper that was published recently on doing this sequential extraction to produce these paired omics data sets, at least for the proteomics and metabolomics. Are there any questions so far? Otherwise, we'll start to get into some of the data now, and I'll explain some of these techniques as we go through. Yes, sir.
As you're expanding the additional cell, obviously you can control a lot of the stuff that goes in your temperature and chemicals you're putting in, whatnot. But how does that compare to what the neighboring cells might put out that would influence, given what's the balance there? Is it mostly under your control, or are the cells growing around a given cell affecting that cell?
It's a good question. It's really, yes.
Can you repeat the question?
Okay, yeah. So the question was, how much of the, we can control some of the process, by in the cell culture format in what we put in the media or what we put in the cells. But then there's also the interacting cells, the other cells within that culture dish that are also conditioning the media or affecting the local environment as well. So how much of that can we control, and how much of that is just determined by the other cells in the dish? And really, it is a mixed bag. So we can control quite a bit, but there's still some signaling going on within the cells between each other that can be basically modulating their internal environment as well. So that's part of the underlying processes that we don't fully understand that we'd like to try to better understand in this process. Does that answer your question more or less?
Yeah, sure.
And was there another question over here?
Excuse me. If you're doing all of this to try and figure out how to tell whether it's a good batch or not early in the process, do you have a sense of how many times this procedure has to be repeated before you really understand what's happening?
Yeah, so for this data set, we're actually just taking 2 batches. So 1 good batch and 1 bad batch. Now we expect there to be variability within what becomes good and even higher variability in how a batch goes badly. But we're using this to try to find initial hits to then do follow-up studies with many additional batches to try to better parse out which of these are actually more definitive and which ones fall out of the initial analysis but don't bear out an additional confirmatory analysis. Does that make sense?
So you're not trying to repeat the good and bad to make sure they match up before you go on.
So for this data set, just because of the scale of the data we're working with already, this is just from one batch of good cells and 1 batch of bad cells at different time points. So each experimental data set is 32 points that's in quadruplicate. So there's 4 samples per time point, crossed into 8 time points, and then we have that for a good and a bad. So we've already got a data set of 64 samples that we're running these multiomics on. Again, that's to look at kind of what we first start to see as going out between the good and the bad, and then doing some confirmatory analyses to see, okay, well, now let's see if these actually hold up in additional analyses. These are also very expensive techniques to be able to do some of these omics level characterization. So first I'll talk a little bit about how we do these protein analyses, and I'll start by introducing mass spectrometry. So here's a diagram from this paper where you can take your initial, so for us, we have cells, or you can take tissues from basically any source of protein. So you could even do this with, your milk, for example, if you wanted to. You extract the protein. So as I mentioned before, we do a methanol-based extraction. So we can add methanol, all the proteins crash out of solution, and we get a nice little pellet. We can then digest these, so we can use specific proteins to go in and actually cut these proteins in our sample up into smaller pieces. So we have these very large proteins, and we cut them down into small pieces. We can then separate these on what's called a liquid chromatography system, or this is a high-performance liquid chromatography system, to basically separate them in time. So we take this very complex mixture that we cut into a lot of little pieces, and this is just to try to get them to separate out in time so that you only have a few of them hitting the instrument at a time instead of hitting all of them on the instrument at one time. It gets a very complex sample. This is just to try to separate them out over time so you can get higher resolution on each individual piece. These then get sprayed into the mass spectrometer, which, to simplify things, essentially looks at the size to charge ratio, or essentially just the size for this case, of those different fragments. You can then select specific fragments or specific of these little pieces of proteins, hit them with high energy to break them into smaller pieces. And then if you break it into enough smaller pieces, you can actually get individual peptides, so individual amino acids that comprise each of these individual peptides. You can then take, if you know what the amino acid sequence is from doing this fragmentation, you can then back calculate what the DNA sequence was, because we know, the codon tables, if you remember from way back when, the codon tables. And then through the Human Genome Project, it's very well curated databases online, so you know what sequence codes for what protein. And then you can essentially back calculate what protein each of these individual fragments came from. So even though you start with intact proteins, you break them into smaller pieces, you go through the instrument, you're essentially trying to go back and calculate what this mixture was to start with. Does that make sense? The other thing we can do is, if you're familiar with the other molecular biology technique called Western blotting, where you can use antibodies, this is a targeted approach. So this is something we'll be doing in some of those confirmatory analyses I mentioned. So we're using this to look at all the proteins we possibly can in the sample. Once we identify key proteins for further follow-up, we can then do targeted studies to look at those specific proteins of interest. So here we're talking, in the order of thousands of proteins that we can analyze. And here we maybe a dozen or so proteins if I want to do a lot of work in validating a lot of antibodies. So now we'll start to get into some of the data. I understand this is a very complicated slide, but first thing we always like to look at is what's called a heat map. So we can look at the differential expression of different proteins. You couldn't see the protein names even if I had them on here, so I don't have the protein names on here. But we can do a clustering And we can see on the across the top here, we've got the different time points, so day 0 through day 16. And then within each of those larger columns of the day, on the left, we've got the bad shown in the blue here, and the good in the red, and the good in the green. And we can see, hopefully what you can start to appreciate here is you don't see much difference in day 0, which we don't expect there to be as much difference in day 0. We expect these to be, as similar as possible. But we start to see significant differences that creep up over time. So even as early as day 2, for example, we see this large batch of proteins that are upregulated in the good differentiation versus the bad differentiation. So one agnostic way we can try to look at these data and try to plot them in kind of the multivariate relationships between these particular proteins of interest is what's called a principal component analysis. So what this does is tries to maximize the spread of the data and collapse it into, in this case, 2 dimensions. So we're looking at each of these different symbols here is the color coding encodes what time it was. So day 0 all the way through day 16. Triangles are for the bad differentiation that resulted in a final purity of 25%. And the filled in circles are for the good differentiation that resulted in a final purity of 75%. This is an agnostic technique, so we don't provide any of this information when we're creating this graph. So it doesn't know what sample is what. It just knows it's trying to maximize the spread of the data. What we see here is we actually get this difference in time. So if we look at principal component 1 here, which explains 28% of the variance in this entire data set, it actually regresses with the time. So we see early time points have a low score on this principal component, and late time points have a high score on this principal component, showing kind of this overall trend from early time points to later time points that's explained in this data set. Does that make sense more or less? So now we'll talk a little bit about how we're starting to analyze. So that's just kind of one kind of global view on how we can start to look at the data, but it doesn't really tell us much about what's driving these differences. For this particular 2-factor experiment, because we have condition, good and bad, and time, we essentially have all these different groups that we're analyzing all the way through day 16. So there's actually 8 time points, but I've collapsed it down to just these 4 for each one for simplicity. It looks like we're dropping off again. So we have all these different groups that we're trying to compare differences over time. So the first way we're going to look at this data.
So you're still.
Is it still showing?
Yeah.
Okay. The first way we're going to look at this data is how are things changing over time in a good and a bad differentiation, and compare those. So just looking at, if you're familiar with statistical.
Just a 12.
Yeah, it's still waiting on my end to relaunch. Should I continue or should I wait for?
Yeah, if you can keep talking while
Okay.
it reboots, that'd be great.
Okay. So we're looking at, if you're familiar with statistical techniques, we're using what's called an ANOVA, or an analysis of variance, to try to see what's different over time generally, and the good differentiation and the bad differentiation. And then comparing what we see changing over time between these 2 data sets. So here I'll show you a few different slides on these are protein abundances. And to orient you, on the left for each of these plots, I'll have the bad differentiation with that low purity of cardiomyocytes. On the right plots, we've got the good differentiation. And then on the y-axis here, we have the relative expression. So this is actually log 2 of the total abundance of that protein in the data set. Now it'll let me relaunch. Okay. So up here, so and then for each plot is a different time point. So we've got all the way from day 0 all the way through day 16 at these different time points through the process. So on the top here, we've got CTNT, which is cardiac troponin T. So this is our gold standard marker for cardiomyocytes. So we expect it to be turned on in a cardiomyocyte, and we expect it to be off in a non-cardiomyocyte. And what you'll see here is we do indeed have higher expression of this particular protein, of our cardiac specific protein, in our good differentiation versus our bad differentiation at the end, which is what we'd expect since we have more cardiomyocytes in a good versus a bad, right? And in fact, we see a 3.2-fold higher expression level of cardiac troponin T at our final time point in the good versus the bad, which actually corresponds quite well with our 3-fold higher purity of cardiomyocytes in the good versus the bad differentiation. Another protein that we expect to be upregulated in a bad differentiation is collagen 1, which is secreted by a lot of potential off-target cell types. So it's a matrix protein, so it's a protein that cells secrete to try to modulate their local environment. And it's quite heavily secreted by fibroblasts or skin cells and things like that. And these are potential off-target cell types in the differentiation. And here we do see, quite significant 32-fold higher production of this collagen in the bad differentiation versus the good. Again, the cardiomyocytes shouldn't be secreting this, so this is likely coming from those off-target cell types, because we still do have 25% of our cells are not cardiomyocytes in this differentiation. So that's good to see, kind of confirms that the data looks good, and that it's at least it's showing what we expect to see. The other interesting thing we start to see in these data sets is if you look at the different time of onset of these particular proteins. So cardiomyocyte T, again, is a protein that we expect to be on in cardiomyocytes and often non-cardiomyocytes. And we actually see it starts to turn on, if you can appreciate, by day 6 here, so 0, 2, 4, 6 in the good differentiation. But it actually starts to turn on earlier, as early as day 4 in the bad differentiation, although the final purity or the final amount is much higher in the good differentiation as we expected. So kind of an interesting temporal change that we're starting to see there. Collagen 1 is a very similar story. It starts to turn on earlier in the bad differentiation versus good differentiation, suggesting potentially there could be some temporal deficits in the differentiation or kind of earlier signaling cascades that are occurring, or signaling cascades that are occurring earlier in a bad versus a good that could be leading to some of this aberrant signaling and aberrant differentiation. These, I'm not gonna go into too many detail on what these specific proteins are, but if we do that statistical analysis of the differences over time, here are a few other proteins or a few different example profiles we start to see as different between the good and the bad differentiations over time. So for example, here's a protein that we see gradually turn on the bad, but has this basically stark increase and leveling off in a good differentiation. We see some other ones where you have kind of delayed dynamics in the good versus the bad. And some other ones that we see that are continually secreted over time in a good differentiation or continually depleted over time in a bad differentiation versus being essentially standard or essentially level within a good differentiation. I say all that to just say, we are starting to see some potential trends that we can see very early in time. So much earlier than our, normally we wouldn't know until day 13 or day 16 if we have cardiomyocytes. We are starting to see some potential trends that we can detect early in time that could be predictive of the final cardiomyocyte purity. So that's essentially what I'm just saying here. Another analysis, where are we on time since I don't have, there's no clock in here. Oh, there we go. Okay. So another analysis we can do is what's called a network analysis. So to roughly orient you to this, we can take all our proteins and we can take known interactions between these proteins, and we can develop what's called a network. And so basically, it's taking any protein and saying what other proteins that we detected in the data set are related to that protein. So for example, if one protein modifies another protein, that'll have an edge on it, or it'll link between these two different proteins, where each of the circles is a protein. And what we can do is we can look at what's called the degree or the number of other things that relates to, and the betweenness, which is essentially how often this comes between the shortest path between other ones. So this one, for example, has high betweenness, since basically any of these proteins to get to any of these proteins have to go through this one intermediate. And these can start to give us some hints as to kind of master regulators in this process, or proteins that are particularly important in distinguishing between the different groups. So here's an example, and I don't expect you to be able to read this all, but here's an example of what one of these networks looks like for our data set, where you can see there's particular proteins that have a high degree or a high amount of connectedness between other nodes, suggesting these could be very important in the process. And then I'm not going to go into these in too much detail, but essentially we can do this analysis, and we can look at we can start to identify these particular proteins that might be very, very important in this overall signaling cascade. Does that make sense?
Aaron, can I interrupt you?
Yes.
Do you have a?
I do on the left side. Yep.
Let's see if this one.
Okay. So what I've done now is I'll be showing the outputs of this network analysis. And now we're looking at just the differences at each individual time point between the good and the bad. So we're neglecting the temporal differences right now for the data that I'll show. And we're just looking at day 0. If we just look at day 0 when we analyze good and bad, what are the differences that we see in how these networks look between a good differentiation and a bad differentiation, and then for the other time points as well. So we can look at here, just looking at I'll say day 0, as we mentioned on that heat map, we didn't see any differences at all. Okay. We didn't see any differences at all, so I'm not showing those here. So we just start with day 2 and then day 4. And so this is day 2, good divided by bad, wherein blue is upregulated, so it's higher in a good differentiation, and red is downregulated, or it's lower in a good differentiation or higher in a bad differentiation. And this is just looking at a lot of different, looking at the outputs of those nodes that are particularly important in this process. And we're starting to identify particular signaling cascades or particular proteins of interest that can distinguish as early as day 2 between a good and a bad differentiation in this data set. So here's just the ones from day 2. And then we see, day 4 and day 6 as well. And there are some that are conserved over multiple timescales. So for example, this protein EP300, which is actually a protein that modifies the DNA status, or how the DNA is folded within the nucleus, is consistently upregulated at early time points in a good differentiation versus a bad. There's other ones, a lot of collagens, which I mentioned, are secreted by a lot of these off-target cell types, as we would expect to be upregulated in a bad differentiation, are indeed continually upregulated at later time points in the bad differentiation. I don't expect you to understand, or I don't expect you to know what any of these proteins do. A lot of this analysis is still ongoing. We're still trying to investigate more of these and perform a lot more analyses to try to identify key candidates within these data sets. A couple of different proteins that I'll look at that were particularly interesting from this analysis and another analysis we performed are, as I mentioned, this EP300 protein. And why this is particularly interesting is here the scale is a little bit different. So day 0, this is just looking at day 0 and day 2. The bad ones are on the left. So day 0 for the bad, day 2 for the bad, and then day 0 for the good, day 2 for the good. And we see basically these opposite trends in the expression patterns, wherein they both start at very similar levels, although there's a little bit higher variability in this particular data set for the good. But we see essentially this pathway, or this protein, being downregulated from day 0 to day 2 in a bad differentiation, and upregulated from day 0 to day 2 in a good differentiation. So this is a particularly interesting protein that it warrants further investigation in some of our ongoing studies. Another one that shows essentially the opposite trend, upregulated in a bad differentiation and downregulated in a good differentiation, as this other protein, NDRG3. Again, I don't expect you to know what this protein is. The last type of analysis that I'll show here for this 2-factor experimental design is we can look at what changes in a good differentiation from day 0 to day 2, what changes in a bad differentiation from day 0 to day 2, and then compare that. So now instead of looking at just day 0 versus day 0 or day 2 versus day 2, we're looking at what is being upregulated in time for a good differentiation, and how does that compare to what's being upregulated in time for a bad differentiation. And again, we can do those pairwise comparisons for later time points as well. Here again, I'm not going to belabor the point, but we can develop very similar graphs, and we can see, for example, again, we get this EP300 protein that, as I mentioned on our previous slide, is upregulated in a good differentiation and downregulated in a bad differentiation. So here we're really trying to find ones that are differentially expressed between the good and the bad. So if they have very similar trends, so for example, over here, this particular protein, we see upregulated, almost 3-fold in a good differentiation, and also upregulated almost 3-fold in a bad differentiation. That shows us that's probably not very specific to cardiomyocytes. It's kind of an earlier process that's going on, but it's not specific to our process of interest. So that would kind of eliminate that as a candidate marker to follow up on. So overall, kind of the conclusions on the proteomics side of things, and I'll go quickly through the other stuff. I go most in depth on the proteomics. That's where most of our analysis has been focused so far. We're currently working to try to rank these top candidates. So obviously, I've shown you maybe 100 or so odd proteins here of our data set of thousands. But we need to kind of further winnow these down to try to get a key set of proteins that to start doing further investigations to kind of confirm if these are conserved across multiple differentiation batches. Some challenges that we've been facing in this experiment, in the analysis in particular, is 2-factor statistical pathway models are not well established. So doing these network analyses really only work for comparison of A versus B, or 2 groups. As I mentioned here, we've got 16 groups. So a lot of the ways we have to end up analyzing these are you do a lot of pairwise comparisons. So as I've shown here, day 0 versus day 2, day 0 versus day 0, and the good versus the bad, things like that, to try to condense the data down. So it's very, very complicated to try to analyze these and then to follow up on analyses. The other thing is proteome coverage and missingness complicates the analysis as well. So we call it proteomics, and that we're trying to identify every protein in the cell, but we can't identify every protein in the cell, particularly low abundance ones or ones that don't digest well or don't break into small pieces very well. So it makes it hard. There's a lot of proteins that might be very interesting that we're not able to detect with this technique. So then now I'll talk briefly about the metabolic fingerprints, or this is the intracellular metabolomics, the metabolites within the cells. Again, we can look at the heat map and we can see some differences. As early as day 2, we start to see differences segregating between the good and the bad, whereas at day 0, they're very similar. Again, we can look at a principal component analysis. This one doesn't really separate in time, but we do see this interesting separation in principal component 1, which I will say doesn't explain a lot of the variance. It only explains about 18% of the variance in the data. But we do see essentially very low scores of this principal component in the bad differentiation or in the good differentiations versus in the bad differentiations in the triangles here. Again, that doesn't tell us much, and we'd like to look at additional, at what particular markers are driving these differences. So here's an example of one. Again, as I showed the cardiac troponin T and the protein side of things, here's a protein that we expect, or here's a metabolite that we expect to be upregulated in cardiomyocytes. As I mentioned earlier, there's this creatine kinase, this shuttling of phosphates between the mitochondria and the sarcomeres to be able to allow that contraction. And this is heavily important in the cardiomyocytes. So we expect it to be upregulated in a cardiomyocyte. And indeed, we do see much higher expression of this particular protein or this particular metabolite in the good versus the bad, confirming that the data is at least telling us what we expect it to be telling us, such that we can then use it for further downstream exploratory analyses. Again, we see this potential differential onset of expression where creatine potentially has started to be upregulated as early as day 8 in a good differentiation, and not until day 10 in a bad differentiation, suggesting some potential developmental timing aspects that are different between the good and the bad differentiations. As I mentioned on the previous one, just as an example of a few different proteins we're starting to notice as different between the good and the bad differentiations, particularly at early time points, a few different metabolites that we're noticing here that I won't go into too much detail, so I'm running out of time. One that we didn't necessarily expect beforehand, but makes sense to us, is this carnitine. So I mentioned that looking at fatty acid metabolism, which we expect to be upregulated in cardiomyocytes versus the stem cells at least, that one of the key metabolites in getting these fatty acids into the mitochondria to be able to utilize them is carnitine. And indeed, we do see an increased production of, or an increased intracellular content of carnitine in the good versus the bad differentiation scale. So although we didn't necessarily predict this beforehand, it does make sense to us. And kind of the data is telling us that it makes sense. And overall, kind of we're seeing some early and intermediate metabolite signatures that could also be predictive of this overall trajectory. Ongoing work, a lot of work, as I mentioned, for the proteomics as well. So trying to identify key candidates for follow-up experiments. A lot of the same challenges for the proteomics. There's missingness in the metabolites as well. Another big problem with metabolites is the databases for the mass spectrometry databases for identifying metabolites are not as well established as the proteomics. Because proteomics, you can go back to the human genome, which is well understood and which is well mapped out. Metabolites are a little bit harder to identify. And there's not really like a genome or something you can go back to identify what all potential metabolites you might have in your system. And the confidence in metabolite is not as high. So there's a lot of metabolites that have a very similar, they can show up in a very similar region on the spectrum of your mass spec. So it's hard to give definitive identification. You might get, okay, well, we know this particular feature of interest could be either, it could be one sugar, or it could be either glucose or fructose, right? Which look very similar. And then finally, I'll briefly talk about metabolic footprinting. So this is looking at cell culture media, or the extracellular metabolites. I mentioned we use NMR, nuclear magnetic resonance, for this, where we can just take the cell culture media, run it on the NMR. And essentially what we're looking at here is differences over time. So the cells are exposed to this media for a certain duration of time. So at the initial time point, we have basically everything that we put into the media is there. So we've got, glucose, amino acids, a lot of other things for all the different metabolites that are present within the basal media formulation. Over time, the cells condition this, so they consume nutrients from that media, and they also secrete byproducts into that media. And we know the time from when we put the media on to when we take the media off, so we can actually calculate a delta over time. In case you're interested, the way nuclear magnetic resonance works is essentially it looks at the nuclear spins in our case, for hydrogens. So each of your molecules that has hydrogens on it will induce basically an oscillation on the coil. When we hit it with a magnet, and then we can detect basically the frequency at which the hydrogens are spinning on these on the different molecules. And then we can convert this highly convoluted spectrum. So you can see it's basically a lot of overlaid sigmoidal profile or sinusoidal profiles, and we can transfer it into the time domain or into the frequency domain to get kind of what, if you remember way back to organic chemistry, looks like a traditional spectrum. And again, if you remember back to organic chemistry, depending where these particular peaks show up on the spectrum, gives you an identification of what particular part of a molecule that hydrogen is attached to. So you can, by understanding where the peak lays down in the spectrum or appears in the spectrum, you can identify what metabolite it came from or is likely to have come from. Obviously, these are highly convoluted spectra, so there's hundreds and hundreds of metabolites that are present within the cell culture media. So you get these very convoluted spectra, and as bad as identifying metabolites from the mass spectrometry is, it's even harder for NMR. It's a lot of this is very manual annotation and manually trying to identify a particular metabolite species. I'll skip through some of these. We did some initial analyses to make sure that we could see differences between good and bad at the end of the differentiation. So essentially between cardiomyocytes and non-cardiomyocytes. And we saw that, yes, we could indeed detect differences between non-cardiomyocytes and cardiomyocytes. We also did an initial run of just a good differentiation to see, are there differences at early time points versus late time points? And we do see these interesting trajectories that occur on a principal component analysis. But then what I'll show here is we did this experiment for good and bad differentiations. We also added in an intermediate differentiation. It's a lot easier to collect these samples, and to run these samples is a lot more cost effective. So we have here in the magenta are bad differentiations, green are good, and then in yellow are intermediate differentiations. Hopefully you can appreciate decent distinguishing between the good and the bad, although the intermediate kind of limits our discriminatory power. But I show this to say what we can do is, if we see some differences, for example, here we see component 1, which explains a high % of the variance in the data, distinguishes decently well from the bad and the good differentiations. We can go in and see what particular part of the spectrum is driving this difference on that principal component. And then now, instead of that very highly convoluted spectra that I showed earlier that has a lot of peaks, we can go in and say, okay, instead of trying to manually identify every one of those hundreds and hundreds of peaks, we can focus our efforts on just these ones that actually drive the difference. So there's a lot of other peaks down here and a lot of peaks up here that we don't care about, since they're not actually different between our good, our bad, or intermediate differentiations. So what we can do is we can go through and we can manually annotate these. So here's an example of glucose, which has a lot of different peaks on it. And we see here's our spectrum that we see a lot of different peaks for glucose. So most of these are overlapping with glucose. There are a few other ones. For example, this one here at 3.35, I believe, that is not part of glucose. It is a different metabolite that we're trying to identify as well. And you'll see these are basically all of these peaks that are downward here in the blue. Similarly, we can go in and we can identify lactate, which are these two peaks in red. Here's the database spectrum for lactate, and here's our spectra, which overlap with these two particular regions on the spectrum. So we can go through and we can once we've annotated those metabolites, we can now then go through and do a quantitation on these, and we can look at different trends in those over time. Here we do see, because we're subtracting out that initial cell culture media, which has a lot of glucose but no lactate. When we subtract that out, we get basically a decrease in the glucose signal. Means we're using more of the glucose, so it's depleted in the cell culture media because the cells are using more of it, if that makes sense. And then lactate, a higher level, means it's increased secretion into the media. So there's no lactate to start with, but we continually put more and more lactate into the media. So similar ongoing work on manual spectral processing and trying to definitively identify these particular metabolites. A lot of similar challenges. There's lower, the limit of detection or the sensitivity of NMR is much less than mass spectrometry. So we get many fewer metabolites. So for proteomics, we get maybe 4,000 or 5,000 proteins. For the intracellular mass spec-based metabolomics, we get maybe on the order of 1,000 or 2,000 metabolites. For NMR, we might get maybe 100 or 200 metabolites coverage. And then, as I mentioned, the metabolite identification is actually much more complicated and much more manual process. I'll skip this since I see we're essentially out of time. The main takeaway here is just a case study of trying to combine the proteomics data, the NMR data from the exometabolomics or the metabolic footprints, and the intracellular metabolite data, just looking at a particular pathway of glycolysis. We can start to see conserved differences over time between these different data sets, and potentially identify surrogate markers. So for example, here, at day 4, we see a lot of proteins that are upregulated, glycolytic proteins that are upregulated in a good differentiation versus a bad differentiation. And we also see that this corresponds quite heavily with a lactate signal, so increased lactate being produced or being detected in the media itself, potentially suggesting that, instead of trying to identify proteins, so you have to collect the cell to get the proteins out of it, we can look just at the cell culture media, which we don't have to disturb the cells at all, to identify that that pathway is upregulated. Obviously, this one makes sense. It's a very well-known pathway, but we're trying to do these types of analyses and identify potential surrogate markers that we can detect in the media that correspond to internal changes in overall development, in overall pathways. And this is my last slide here, just, ongoing work. As I mentioned, we're hoping to get the RNA-seq data back soon. A lot of ongoing analyses on all these data sets. As I mentioned, these are fairly recent that we've collected these data and trying to do some more of those single omic analyses and a lot of integrative analyses. Future experiments, as I mentioned, trying to confirm. So once we identify particular proteins and metabolites of interest, trying to confirm these in additional differentiations, good and bad, and also trying to capture intermediate ones and see if they actually, where they distinguish. Is it like a linear distinguishing where a low level is bad, a high level is good, and in an intermediate level, it means it's going to be an intermediate differentiation? Or is it more binary? Or if it's over 50%, you get a high level. If it's below 50%, you get a low level, for example. Also, some of these regions, particularly this day 2 to day 4 region, we see is very highly dynamic. There's a lot of changes that we're noticing in here. So we're looking to do some finer time resolution sampling. So instead of doing every 2 days, doing every day or maybe every 12 hours to get some more time resolution in that region to see how the dynamics play out there. And then finally, closing the loop, as I mentioned, the initial reason we're doing all this is to try to optimize the overall process. So as we start to identify pathways that are turned on in good differentiations or turned on in bad differentiation, we can then exogenously, through either small molecules or through direct, through like genetic means, we can try to turn on the good pathways. So for example, if we see glycolysis is highly predictive of a good cardiomyocyte differentiation at early time points, we might go in there and say, okay, well, then we would hypothesize if we turn on glycolysis at day 2, maybe we can drive a differentiation more towards a cardiomyocyte versus a non-cardiomyocyte, just as an example, or turn off bad pathways. So that if we see glycolysis is bad, we can say, okay, well, let's go in and inhibit glycolysis. Let's not let the cells do that and hope that we can kind of hit a feedback loop to try to get these cells on a direct path. Some additional perturbations on the overall processing parameters, and then eventually trying to develop methods to do feed forward or feedback control. So if we see at time point 2, we have this particular, so for example, we see at day 2, glycolysis is turned on heavily. We could say, okay, well now instead of failing the batch, let's go in and now let's inhibit it to try to bring it back on specification. And that's all I have today. So I'd like to thank my lab, my PI. The multiomics work for the proteomics and the intracellular metabolomics has been done in collaboration with Dr. Guh here at the University of Wisconsin, over in the cellular and regenerative biology department. And then the NMR spectra has all been, I run these, but through the NMR core here at the University of Wisconsin. So pretty much all of the analyses and even the RNA-seq is being done all with resources here at the University of Wisconsin, in addition to our work with the research center that I mentioned before. I'll take any additional questions that anyone has. Thanks for bearing with me, and for those of you on Zoom, thanks for bearing with the few drops that we had in the presentation there.
Thank you. Network analysis.
Yes.
Seems like lately, it just seems to be popping up in pretty much, every discipline. I kind of look into like, economics, computer networks.
Yeah.
All over the place. Is this kind of like a standard thing now in organic chemistry that people look at
It's becoming more and more utilized. There's a lot of different ways that you can perform a network analysis. So you can perform network analyses just based on correlations between things. So you can say, okay, if you have a large data set, which that's why a lot of the networks are coming out now, why networks are being utilized a lot more, is it helps you to condense very large data sets. And as you know, the world is moving towards larger and larger data sets for everything, for better understanding, being able to understand and visualize and conceptualize these things lends itself to these network analyses. But essentially for large data sets, you can look at correlations. So you can say, every time this particular metabolite goes up, this other metabolite or this other protein goes up. And you can try to construct them based on those interactions, or you can use what we're using here, are curated databases. So of known interactions. So for example, protein A inhibits protein B, and that'll draw an edge between those two proteins. So you can use those to try to identify and understand those a little bit better.
Yeah, it's like, even though all these fields are so different, there's kind of a common mathematical
Yeah.
core.
Yeah,
that
exactly.
applies to all the areas. To me, it seems pretty
Yeah.
fascinating.
And actually on that network analysis slide, the intro slide, on one of those, I didn't go into it, but there's one called PageRank. And that was actually the original algorithm utilized by Google in the search engine to try to rank which one is your top entry versus your last entry. And I actually use an iterative technique to look at, you know, this protein relies on that protein, and there's these other 10 proteins that rely on that protein. So it tries to basically weight that protein as more important because it's got a lot of more connectedness with other proteins. It's a very interesting technique that's been utilized more and more, as you mentioned, in a lot of sets. Other questions? Yes.
How long do you have left in the PhD?
That's a good question. So I am currently a 5th year. These data took a little bit longer to generate. I expect about another year or so to be finishing up here. So it'll be right around 5.5 years, which is actually for our program is fairly fairly average. We in the biological science or in the bio side of the chemical engineering department kind of bring that average up a little bit more, especially the stem cell work, because as I mentioned, this differentiation takes 16 days to get to our final time point. So every experiment you do, 16 days before you know the outcome and before you can design your next experiment. So we try to get creative with a lot of different projects going on, and we try to nest them. But still, the timescales for progression of one experiment are quite long. Any other questions?
Thank you very much. Outstanding results within another year, let us know. We'll listen in again.
Yeah, that's the hope. Okay, so once we get the RNA-seq data back, or the transcriptomics data, hopefully that has a lot. So I mentioned the proteomics is like 4,000 to 6,000 proteins. The transcriptomics data has around 20 to 25,000 different transcripts you can get. So you get a lot more data, you get a lot more overall depth or breadth of the overall data that you're able to analyze there. So hopefully once we have that back, we'll be able to integrate that with these data sets and get a lot more progress going on this project.
So next week, we're back to manure.
Okay.